So, what brings you here today, Mrs. S?
Doctor, it seems that I can do anything but produce insulin.
Don't worry. We are working on a new solution to treat your problem.
Yes, one day I'd love to say that I used to have diabetes.
One of the novel approaches to tackle type 1 diabetes is the synthesis of immune-evading pancreatic cell clusters. Let's begin with what diabetes mellitus is. When we eat food, our body breaks it down into glucose that enters our bloodstream. A hormone called insulin helps our cell absorb glucose from the blood to provide energy. People with diabetes don't produce insulin, or it doesn't work properly. So the blood glucose level increases, causing severe damage to the body's organs. Diabetes can be diagnosed by checking blood glucose levels using the fasting plasma glucose, or the FPG, or the hemoglobin A1C test. Some of the symptoms include extreme fatigue, feeling thirsty, slow healing of wounds, and weight loss.
Statistics from the International Diabetes Foundation estimate the number of diabetic adults in the world to rise from 537 to 783 million by 2045. The UAE is currently ranked the 10th highest in the world for diabetes prevalence. And 1 in 5 adults are estimated to be diabetic according to the Dubai Health Authority. Current treatments for type 1 diabetes include insulin injections, which often present as a costly and chronic burden for patients and do not entirely eliminate the risk of chronic complications associated with diabetes. Transplants of pancreatic beta cells that make insulin may provide a cure, but they require patients to take long-term immunosuppressants. Thus, the generation of insulin-secreting beta cells with immune-evading ability could provide a promising solution. We conducted an extensive literature search of peer-reviewed articles in databases such as PubMed, Google Scholar, and ScienceDirect with the keywords diabetes mellitus, islet transplantation, and the role of PD-L1. The search process uncovered 45 peer-reviewed articles from 2001 to 2021.
Let me take you deeper into each stage of this multi-step process of immune-evasive islet transplantation. First, human-induced pluripotent stem cells are obtained from the lab. Then, a genetic switch called the estrogen-related receptor gamma, or simply ERR gamma, is introduced. This genetic switch turns on certain genes that stimulate insulin production from beta cells in response to glucose. Next, we add 2 important cells, the human adipose-derived stem cells and human umbilical vein endothelial cells. These act as pancreatic fibroblast and endothelial cells respectively to form organ-like and vascular structures in a three-dimensional culture. And why do we need this organ-like or organoid arrangement? That's because pancreatic islets are highly organized cell clusters that have crosstalks between beta, alpha, and other islet cells. Subsequently, we add a protein molecule called WNT4. WNT4 causes the maturation of the beta cell by upregulating mitochondrial genes and, as a result, increase the metabolism of the cell, leading to improved insulin secretion. The most important step is the addition of interferon gamma to induce endogenous programmed death ligand 1, or simply PDL1. PDL1 causes apoptosis or death of local T cells, thereby preventing graft rejection and thus providing the transplanted islets with the ability to evade the immune system. Finally, the islet cell cluster is transplanted into diabetic mice to maintain blood glucose levels. The first figure shows the transplantation of islets expressing PDL1 under the kidney capsule of diabetic mice. The second figure indicates insulin promoter-driven green fluorescent protein expression 27 days after transplantation. This means that when the cell produces green fluorescence, we infer that the cell expresses the target gene, which is insulin. Electron microscopy further reveals insulin and glucagon granules in beta and alpha cells of WNT4 treated organoids, which are structurally very similar to human islets. The graph indicates that the function of organoids lacking PDL1, which is indicated by the red line, was progressively lost as seen by increased blood glucose levels, while organoids expressing PDL1, indicated by the blue line, maintained blood glucose for more than 50 days. In conclusion, immune-shielded stem cells could serve as a potential cure for type 1 diabetes, but more high-powered preclinical research needs to be done to ensure the long-term effects and safety of this procedure before advancing to human trials. Furthermore, research studying the optimal sites of transplantation is still underway. As of now, the liver and the kidney capsule are the preferred sites of transplantation. Thank you.